Very happy morning, students. I hope you're all safe and sound at home. Welcome to RGR Academy. This is a biology session. Let us have a quick recap of what all things that we discussed in the last class. Fine. The last class we discussed about the unit. We completed the unit, the plant physiology. Almost the 5 chapters we done in the plant physiology. That is your photosynthesis, respiration, mineral nutrition, and we discussed about plant growth and regulators, the hormones that we discussed, and at last we discussed about the transpiration and all these mechanisms that we discussed, like stomata, all these things. So by that we come to an end of the particular unit, that is plant physiology. All right. So with that we move to the next part of your biology session, right? Here we start with the biological classification. So as you know, the biological classification. So what were the things that we're going to discuss in this particular unit, the biological classification? As biological classification is like one of the important the unit that has been prescribed as far as Neet syllabus, and the weightage also is quite more next to the plant physiology. So we are in position to discuss about this particular unit, that is biological classification, that is diversity in living organisms. So we start with the diversity in living organisms. First we start with the groups, the organisms like bacteria, virus, fungi, algae, protozoa, plants and animals. These are like one by one we're going to discuss in this particular unit. Before that, like we'll start with the introduction of the classification. We'll start with introduction of the classification, and where how these microorganisms are classified. We have the kingdom concepts, usually we call it as. We have a two kingdom concept, three kingdom concept, four, five, and six kingdom concept. Like until then, we have like different types of kingdoms that we're going to discuss. And also we're going to discuss the characteristic feature of these kingdoms. And after once we're done with this, the classification outline, then we move on to the next part, that is the virus. So as far as the virus is concerned, as you guys know, like virus is not a cell. Virus is just a particle. But instead, even though it's a particle, we categorized under the group of microorganisms. The reason because the virus do show the symptoms and the clinical manifestations comparatively more than that of the other group of microorganisms like bacteria, fungi, algae, protozoa, right? So due to that particular reason, right, so we first start with the virus, and there is no particular place for the virus or room for the virus in the five kingdom or six kingdom classification, even which was proposed by Whitaker, right? But there are like justification that we're going to give, like why, even though the virus is not a cell, why we are talking about the virus under the group of microorganisms. First we discuss, and before we go into the virus in this particular session, we'll begin with the classification of the the living organisms, classification of organisms. First, we'll start the introduction, and then we move on to the next part. Okay. So let us begin with the biology session today. So happy morning to one and all. So I hope you're all like stay connected with us. Thank you guys. Fine. Now we'll start with the first part, that is biological classification. Coming to the biological classification, let us start with uh, okay. All right. So you can, when we just try to connect the virus with the on the particular session, you can ask questions. Of course, you can ask the questions, but related to the subject, you're most welcome to ask the questions. Okay. So guys, you please refrain from asking questions irrelevant to the subjects, whatever that we will be discussing. Okay, so that we can save our time as well. All right, guys. We'll start with the diversity in living organisms. We'll start with bioclassification. Bioclassification. So let me start with the introduction of this bioclassification. Once we're done with this, we move to the other parts. So this bioclassification, which was first proposed by Carolus Linnaeus. This is a well-known fact. Like he's the person first. He's the one. This is L. Carolus Linnaeus. Carolus Linnaeus is the first person who identified or classified the biological organisms. Like he made a classification. So we discussed first about the biological classification, and he termed as father of taxonomy. Carolus Linnaeus is called as father of taxonomy. Father of taxonomy. Fine. So, as far as system is concerned, when we talk about the biological classification, we have three systems. So, what are the three systems? We have artificial system, natural system, and we have phylogenetic system. So, the systems that we're talking about is artificial system, natural system, and we have phylogenic phylogenetic system. We have three types of system on the whole we have. So this artificial system, which was the chapter is classification, the diversity in living organisms that we're talking about, diversity in living organisms. Okay. As far as the artificial system is concerned, this began in the 18th century itself. This began in the 18th century. So, what type of questions that you get, especially with the system is concerned? The genera planetarium. Usually, this type of questions, the genera planetarium, planetarium, genera planetarium. Okay, so bioclassification is the chapter that we're talking about. So genera planetorum, and this was proposed in the year 1737. 1737. Okay, so genera planetorum and species planetorum. Species planetorum. Species planetorum. These species planetorum in the 1753, and this type of classification is called a sexual system of classification. This we term it as sexual system of classification, and this was even proposed by Aristotle. This type of classification, which was proposed by Aristotle in the very beginning, we're talking about this classification. It's a Aristotle. So this is a sexual system of classification. And we have before that we have a Theophrastus, we call it before even Aristotle. We talk about the first person who begin with this artificial classification is Theophrastus, and then comes like Aristotle, which began in the 18th century itself. By means of this publications, we could see in Genera Planetarium and Species Planetorum. It's issued in 1753. So from this part, you can get a question. Genera Planetorum and Species Planetorum and the year. This one area you can get a question. A natural system of classification. This is like similarities into the relation to the progenies that we're talking about. Natural is similarities. This in terms of similarities in relation to progenies. We're talking about the natural system of classification. This proposed by Carolus Linnaeus. This we call it a system natural. So this was proposed by Linnaeus, system natural. We call it a system natural, which was proposed. The natural system was classified by proposed by Carolus Linnaeus in the year 1758, and then comes Bentham and Hooker in the Genera Planetarium, and then comes Bentham. So you need to remember the name of the scientist here, Bentham and Hooker. So as far as natural system of classification, Carolus Linnaeus and Bentham and Hooker. Bentham and Hooker in the year 1863. Then this comes like 1863. Fine. So this is about you need to remember the names here. The Carolus Linnaeus for system natural, Bentham and Hooker for the Genera Planetorum in the year 1863. Okay. So this weightage not from this particular chapter, as far as like complete the unit is concerned, as far as like diversity in living organisms, the weightage is like you can get 16% of questions that you can get from this particular chapter. Maybe next session I give all the chapters, how much of weightage that will be given for all the units. In the next session, I'll disclose all the unit wise and chapter wise weightage as far as Neet is concerned. All right, fine. So next to the plant physiology, so I am plant physiology, animal physiology. Next to that, you can give importance for the diversity in living organisms as far as botany is concerned. Okay, so artificial system of classification, natural system of classification, and come to the phylogenetic system of classification. Phylogenetic system of classification here, which talks about the genetic relationship, evolutionary and genetic relationship that we're talking. So on the whole, the bioclassification is based on three things, parameters like artificial, natural, and the phylogenetic system of classification. Now we move to the detailed classification where as far as detailed classification is concerned. As you guys are aware of the two kingdom concept, three kingdom concept, 4 and 5, and six kingdom concept. So only thing I just want to tell you is like who proposed two kingdom concept. Okay, okay. So as far as Bentham, you asked Bentham and Hooker is 1863. 1863 for Bentham and Hooker. So two kingdom classification. So three kingdom classification, four kingdom, five kingdom classification, and also we have six kingdom classification. So who proposed two kingdom classification? Carolus Linnaeus. Carolus Linnaeus. Linnaeus is the person who proposed two kingdom classification. He just classified just into plants and animals. Just only there was two kingdom. As far as two kingdom classification concerned, nothing much like there was no Monera, no Protista, there's no from no room like other group of organisms. It just classified just like plants and animals. So here like only two kingdom classification by Carolus Linnaeus in the year 1758. Guys, this area can get a questions as far as scientists are concerned. Carolus Linnaeus proposed a two kingdom concept and just classified only the plants and animals. Coming to three kingdom concept, who proposed three kingdom concept? Ernst Haeckel. Ernst Haeckel. Ernst Haeckel proposed three kingdom concept. And coming to the four kingdom concept proposed by Copeland. Copeland. Five kingdom concept proposed by R.H. Whittaker. R.H. Whittaker. And six kingdom concept which was proposed by the scientist name Oos. So from any part you can get it, because most commonly we discuss only about the five kingdom concept in most of the textbooks, but also you should understand the proposals, right? Who proposed, right? Two kingdom concept by Carolus Linnaeus, three kingdom concept by Ernst Haeckel in the year 1866. 1866, Ernst Haeckel proposed three kingdom concept, but he included along with the animals and plants, he also included something called as Protista. So here plants, animals, and also here included something called as eukaryotic organisms, like single cell eukaryotic organisms that here included is Protista. So eukaryotic single cell organisms also been included. But in this case, Carolus Linnaeus he just classified the plants and animal, only the plants and animals, like only these two kingdoms which was done. Okay, fine. So two kingdom concept, and then we have the three kingdom concept in 1866. Coming to the four kingdom concept by Copeland. Copeland in the year 1956. 1956 Copeland. Okay, Copeland is a 1956. In this year, he included like animal, plants. Then we have a Protista and Monera. We have like Protista. We have like plants, animals, and then we have Protista, and we have a Monera, and also we have a Monera. So this is about the classification that you could see in case of Copeland. All right, guys. So next, move on to the five kingdom concept. As far as five kingdom concept, which is proposed by Whittaker, we talk about the Whittaker's five kingdom concept, and he proposed in the year 1969. 1969, Whittaker's nine kingdom concept. In this case, as far as Whittaker's five kingdom concept is concerned, there are five kingdoms like Monera, Protista, Fungi, Plantae, and Animalia. So here five kingdoms that he was proposing: Monera, we have a Protista, and we have a separate room for Fungi, and we have a for plant, and also we for a Animalia. So this is was a proposal of R.H. Whittaker. Okay, and coming to the six kingdom classification. So there was only one additional one separate kingdoms which was placed in the six kingdom concept. There was a division of the Monera. So divisions of Monera which took place into two actually. That took place in the proposed in 1758. So are you sure Linnaeus 1758? Can you please reconfirm in the textbooks, guys, like Carolus Linnaeus the year? Because as far as my reference, it shows like 1758, but if you're telling like 1735. So can you please confirm me like 1758 or 1735? So if you give me a confirmation as far as years concerned, I can change it better. Okay. So I given it to you. Okay. 1758 or as you said it is 1735. Okay, Kirti, are you sure it is 1735? Then we can go with 1735 itself. Okay, it's no big deal in that. Coming to the ooze. Ooze is concerned where he just subdivided this particular part, that is Munira. Okay. Then as per your confirmation, I changed 1738 into 1735. So guys, please do confirm. Okay, please do confirm. As per the state book is concerned, you're telling it's 1735. So based on that, I'm changing to 1735. Okay, guys, I'm changed to 1735 for Carlos Linnaeus 1735. Okay, guys. Okay, make this change. And coming to Wu's six kingdom concept where, like, along with Carl Woos and Candler and Willis, there are other two guys like Candler and Willis. So these two, the three guys together, they proposed a six kingdom concept. Okay, six kingdom concept. Coming to this six kingdom concept, where yeah, as far as Carl who's concerned, it's actually they given 1990. They given in the year 1990. Yeah, the year for Six Kingdom concept is 1990. But what's the only inclusion they made in the Six Kingdom concept is they classify the Monera, usually the bacterial species, into two. One is Archaebacteria, very primitive old, that is older bacteria, and other type is the eubacteria, true bacteria. So these two classifications which was made as far as Monera is concerned. So these guys who's made the classification, sub-classification, where he divided Monera into two kingdoms. What are the two kingdoms? One is Archaebacteria groups, that is Adophiles, Dermophiles, this type of Archaebacteria species, and Eubacteria. So primitive type of bacteria and eubacteria. This was the sub-classification which was made by Woos in the year 1990. So this made the sixth kingdom concept. So he made this as the first kingdom, this is the second kingdom, this is a three, four, and this is five, and this one is the sixth kingdom concept. So on the whole, as far as classification is concerned, we have six types of classifications, started from two kingdom, three kingdom. Guys, remember from this area you can definitely one question get a questions like maybe the scientist or maybe the by means of the year you can get a questions. For that one day I'm just giving yeah, archaebacteria are the very primitive bacteria when the hot springs and volcanoes and the very old, like very primitive bacteria at the time of evolution is concerned, that methanogens, allophils, this type of bacteria, how it's originated, like these bacteria that we discussed, that separate kingdom like archae, and eubacteria like other two true bacteria, like where I got the peculiar normal cell walls and what we discussed. So this is being classified as a second kingdom. So this is about the classification, biological classification is concerned. But in the most of the textbooks, like we give more importance for the five kingdom concepts. Coming to the five kingdom concept, which was proposed by R.H. Whitaker. R.H. Whitaker. So we want to the five kingdom classification, diagrammatic representation. Fine. So is Robert Harding Whitaker. This confirmation which was made by this person, Whitaker, that I told you in the year, in the which year it is, in 1969, where you could see the five kingdoms in this place, where Monera, and here we have the Protista, and we have the fungi, then we have animals, and we have plants. These are actually the five kingdom, the classification um, yeah, so that's again like subdivisions actually. Further archaebacteria, that also we can even further divide it, halophils, dermophils, that again you can divide into furthermore. But since we're talking only about the outline of the classifications, I didn't go deeper into the further classification of the archaebacteria. If we started dividing like everything, like we keep on going to numerous branches, then we couldn't complete the session. That's the reason I limited just only with archaebacteria and eubacteria. Non-motile organisms as well as it goes with the asexual mode of reproduction. Second kingdom, this is the first kingdom as far as Whitaker is concerned. This is the first kingdom as far as Whitaker is concerned. Then second kingdom, we're talking about the Protista, right? Protista. See, we are not limiting with like seven kingdom and all those things because again, because the worldwide except globally accepted kingdom is actually five kingdom concept. That itself is stopped with the Whitaker. If you go with the seven kingdom or eight kingdom, if it keeps going, and this is by means of asexual mode of organisms, and it is non-motile organisms. So this is the first kingdom. it's buffering. Give us some time. So yeah. Okay, I didn't start. I didn't do anything. I didn't continue the class. I'm just waiting. Okay, so since it's buffering, I'm waiting. So you don't worry about this. Guys, give me a signal once it's clear for you. From your side, if it's clear, give me a signal so that we can start. Okay, guys, it's clear now. We'll start. Fine. So we are discussing about the five kingdom concept which proposed by Whitaker. Monera, the bacterial species, all the bacterial species that we categorized here. Coming to Protista, you show the diatoms, dinoflagellates, the slime molds, these organisms which comes in the category of the Protista, usually this is of eukaryotic unicellular organisms. Eukaryotic unicellular organisms. Unicellular organisms. Eukaryotic unicellular organisms. And fungi, and fungi also we have the mold as well as the yeast type, right? We have the filament that is fungi and that is like mold as well as the yeast type and animals and the plants. So this is an outline of the five kingdom concept. So Whitaker's five kingdom concept is a confirmatory, the five kingdom concept based on which only further extensions of six kingdoms, the seven kingdoms, the kingdoms that been like increased or been like new inventions can mean. But the basic, the concept which is being given by the Whitaker. So for that reason, we're giving importance for the Whitaker's five kingdom concept because it clearly explained all the main five kingdoms like Monera, Protista, Plantae, Animalia, and Fungi. So this comprises of five kingdom concept and Whitaker, that is full name Robert Herring Whitaker between 1920 to 1980. Okay, so this is about the classifications outline of this of the living organisms. Now, what we're going to do is like as for the five kingdom concept, of course, we discussed about Monera, we discussed about like Protista, the diatoms, dinoflagellates, slime molds. But when we talk about the microorganisms, the living organisms, right, we have the bacteria, algae, fungi, protozoa, we have organisms like plants, we have animals. But you could see there are two things that seems to be missing here. One is your algae, and other one is your virus. You cannot see the particular the position for the virus and algae in this five kingdom concept or any other kingdom that you could see more specific. Can you tell me like why there is no position, there is no particular place for algal group? You could see there is a group separate kingdom for you could see is a fungi can you could see, but what about algae? We can justify virus is not a cell, it's a particle. For that reason only we couldn't categorize under five kingdom concept. But what happened to algae? Why the algae has not been categorized in any of the or we not given a separate kingdom for algal group? Okay, so as far as algae is concerned, algae which comes under like for example, it falls under the group of Monera. It can comes under even the Protista group. Even it can comes under the group of like you could see in the plant group. In all the cases, plants you could see like Protista and also you could see Monera. In all the three kingdoms where you could see the algae which mark its footsteps. Okay, and it's not having a true leaves or stem, but algae which mark with footsteps in all the three kingdoms. You could see the blue-green algae in Monera. Here also you can see the algae. Protista, you could see diatoms in all these like cases. Here also you could see the algae. And also like plants, like in that time, because it's having the chlorophylls and everything, like it's also like perform the photosynthetic reactions. There also you could see the algal species beefing up. So this reason where we cannot the scientists, they cannot give a separate position or the kingdom for the algal group, because algae started sprouting its seeds or wings to all the three kingdoms like Monera, Protista, and the plants in all the three cases where you could see. Yeah, Chromista is a separate kingdom, but this was not given by the Whittaker. And as far as Whittaker kingdom, five kingdom concept is concerned, there was no separate room for algal species, because at that point of invention, the five kingdom concept, if it just categorized algae under, because most of the algal species comes under Monera, Protista, and the plants, and later part only Chromista, the kingdom came actually. The Chromista kingdom came after the invention of the five kingdom concept only, where all the algal groups which has been segregated kingdom. But at the time of five kingdom concept, there was no confirmation, there was no separate kingdom which was given for the algal species. That is the reason why in five kingdom classification you cannot see the separate kingdom for the algal species. This is number one. And second reason thing is virus. Even though we talk about the virus under the microorganisms, right? Virus which cause disease even far better than other type of microorganisms. There is not separate room for virus. So if we start justifying, we start answering like this question: why virus has not been categorized under the five kingdom concept or any other six kingdom, wherever whatever it might be. The reason is virus is not a cell. Anything that we call a plant cell, algal cell, fungal cell, protozoan cell, bacterial cell, we call it as, but nowhere you could see the virus can be classified as a viral cell, because virus is not a cell, because virus doesn't have the structure of its own. It means whatever thing that which has got, it's all received from the host cell, because virus is a obligate parasite and virus is very specific in nature. This property we call it as cell tropism. Cell tropism is very selective. The virus seems to be very selective, and the virus is inactive in the surrounding, but it's active. That is the reason we call it as obligate. Obligate means strict. It's very strict in its host cell. So viruses are obligate parasite, and virus cannot survive often like outside the host cell. So it depends on the host cell, and this dependency is also very specific in nature. So today what we're going to discuss is about, we're done with the outline of classification. Now we'll start with the biological classification. The first thing that we're going to discuss is viral groups. So viral particles will do. Once we're done with the viral group, then we'll start with the Monera and other kingdoms. All right. So coming to the virus, before going to virus, who first discovered virus? Who's the father of virology? Can someone tell me who's the father of virology? So coming to the first part, we discuss about virus. So this is a season of virus. I hope you guys can understand. So it's season of virus. So virus considered as a particles and not a cell. Yes, it's a virus. Virus is a particle. It's not a cell. That is the reason why virus has not been categorized. So initially this virus was actually named as a poisonous. In the Latin terms, like it was termed as it's a poisonous fluid. That was the term which is given for the virus. Virus was termed as a poisonous fluid. And this virus was first actually identified by Louis Pasteur. Louis Pasteur, he first identified this virus group. And after this, actually, the person named Contagium vivum fluidum, who proposed this? Contagium. Sometimes they ask like vivum fluidum. Who proposed this statement? Contagium vivum fluidum. A person who proposed this particular term, contagium vivum fluidum, in the year 1898. 1898. Can someone tell me who is the person who proposed this particular sentence? Okay, yeah, it is Martinus Beijering. Martinus Beijering. He is a person first proposed the statement as virus is a contagium vivum fluidium, MW. Yeah, exactly, in the year 1898. And who is the father of study of virus? We call it as virology. So study of bacteria is called as bacteriology. Study of fungi is called as mycology. Study of algae is called as phycology, right? Study of like protozoans, protozoology, the same way, right? So study of virus, this we call it as virology. Who is the father of virology? So who is the father? So father of taxonomy, we call it as Carolus Linnaeus. The same way, who is the father of virology? Father of virology is W.M. Stanley. W.M. Stanley, they confirm to be the father of Stanley. W.M. Stanley is the father of virology. Okay, fine. So you gives like Bajering and Martinez, but it confirmations given the father of virology is W.M. Stanley. W.M. Stanley is the father of virology. Okay, so don't get confused with the father because lot of inventions that we're going to discuss about the historical perspective. We'll discuss about the historical perspective of the virus. First, we'll start about Ivanovsky. I just give the name of the scientist. Okay, you tell what's his contribution. DJ Ivanovsky. What's his contribution? DJ Ivanovsky. What was his contribution? He first identified the tobacco mosaic virus. Okay, Ivanovsky. And then comes Loeffler. Loeffler and Frosch. FMV virus. They first identified the FMV virus. Guys, this area also can get a question. Okay. Okay. So we'll separately discuss about the coronavirus. Yes, we'll discuss about the coronavirus because it's a season of corona. So definitely you get a questions from the corona also. We'll discuss about the corona also here. Okay. Separate segment we'll discuss. Okay. Definitely virus are smaller than bacteria. So Ivanovsky for the tobacco mosaic virus, Loeffler and the Frosch. Loeffler and the for FMV. What's the FMV? Food and mouth disease, food and mouth viruses, we call it as FME. And Landsteiner popular. And we have what? We have what and Herel. These guys identified the first bacteriophages. Bacteriophages. Okay, bacteriophages by this Stuart and Herel. And we have Anderson and Jacob. Anderson and guys from this area can get a Anderson and guys from this area can get a question because virus historical perspective as well giving information for this one. So Anderson and Jacob, they proposed virion first. They proposed virion. The structure we call it as virion, which was proposed by Anderson, Jacob. And we have what is that process called? Baltimore. Can someone tell me what is the main invention of Baltimore? Baltimore. And this person got the Nobel Prize also. Baltimore. Can someone tell me what's the contribution of Baltimore? Baltimore and the Temin. What's their identification? Baltimore and Temin. They first identified the reverse transcription mechanisms, a reverse transcriptase enzyme. This was first identified by these guys like Baltimore in the year 1970, and they got the Nobel Prize in the year 1975. Okay, this process called as Teminism. This called as Teminism. So this is called as Teminism. Teminism, where they first found a reverse transcriptase. Reverse transcription mechanism. Reverse transcription mechanism, which was proposed by Baltimore and Temin. Okay, so this process called as Teminism. They identified the reverse transcription and they received the Nobel Prize for this invention in the year 1975. Baltimore and these two guys, they received the Nobel Prize for this invention in the year 1975. And Stanley was a person in the year 1982. He first identified prions. The sixth one I write here. Stanley, a person named Stanley. He first identified the proteins. Actually, the proteins not given a genetic material. You would have studied like mad cow disease, Creutzfeldt-Jakob syndrome, right? These are like Stanley, which comes for the prions. And Luc Montagnier is for the HIV virus. Before the Luc Montagnier, there was another person called Robert Gallo. So Robert Gallo, Gallo. Luc Montagnier and Robert Gallo, they first identified HIV virus. So HIV virus was identified by Robert Gallo and Luc Montagnier. Okay, fine. So Herel coined the term bacteriophagy. These are few historical perspective, guys. From this area, you get a question. So for that one, I'm just giving giving importance for historical perspective. Ivanovsky for the tobacco mosaic virus, Frosh and Loeffler, this is for foot and mouth virus. And for Heron, it's for the bacteriophage. Bacteriophage is nothing but the virus which affects the bacteria. And Anderson Jacob is for the virion. And Baltimore and Temem is for the reverse transcription. And Robert Hallow is for the HIV virus. Robert Hallow. And we have Luke Montagner also for the same HIV, and Stanley is for the prions. This is like little bit of information about the historical perspective of viruses. So coming to the viruses, we'll discuss next about the general characteristics of viruses. So when we talk about the general characters, general characters of viruses. Assume the virus is not a cell that we discussed already. Bajering actually proposed like in a contagium vivum fluidum. This proposal was given by the Bajering. Bajering is given the contagium vivum fluidum, and also he isolated most of the viral particles. I mean from the fluids. I mean from the pustules kind of thing, which is on the surface. Like from that he isolated. I mean from the fluids. I mean from the pustules kind of thing, which is on the surface. Like from that he isolated most of the pox viruses. Bajering. Yeah. General characteristic of the viruses, this is ultra microscopic. It's very smaller in size. The smallest is the viral groups. Ultra smaller in size. The smallest is the viral groups. Ultra microscopic. It's very small in nature, and mostly it is intracellular. Viral groups are intracellular. Microscopic. It's very small in nature, and mostly it is intracellular. Viral groups are intracellular. It means it lives only inside the cell, inside the host cell. So it definitely it's a intracellular, and it is smaller in size. And this, since it is very smaller in size, so this can pass through the bacterial filter. Pass through the bacterial filter. Pass through the bacterial filter. So sometimes if you use the bacterial filter also, that cannot filter or that cannot stop the viral particles, because viral particles are smaller than the bacteria. And the nucleic acid, what's the main component of the viral particles? This consists of nucleic acid with what? Nucleic acid, this consists of protein code. This, what is the name of the protein code? We call it as a capsid. So we have the nucleic acid, it may be a DNA or it may be a RNA, but this comprises of the protein code. Okay, and no proper cellular organelles that you could see. You could not see the no proper cellular organelles inside, and also no proper metabolic machinery or metabolic reaction that takes place inside the virus. That also is there. And mostly this virus, which takes two types of life cycle. One is virulent cycle and avirulent cycle. Also we call it as lytic cycle or lysogenic cycle. This go with lytic or lysogenic cycle, or it can take both. So the virus may take either lytic cycle, where the destruction of host cell takes place, right? And lysogenic cycle, there is no destruction, where the viral genome get integrated with the host genome, or it may go with both lytic as well as the lysogenic. This is the two types of life cycle where the viral particles usually it go ahead with. Okay, and this cannot grow and divide outside the cell. So this also main important features. It cannot grow. It can't grow outside the cell. Outside the cell. It means if you want to culture this particular virus also, like you cannot go for the normal medium like mutant agar, the normal medium that you give for the bacteria, that is not quite sufficient for the growth of the then how we can cultivate the virus? You can cultivate by means of, can someone tell me, if you want to cultivate the viral groups, then since I told you like it cannot grow outside the cell, so what type of like provision, what type of like nourishment that you can give for the virus so that the viral particles can grow? Usually the viral particles, we prefer like tissue culture. We have egg inoculation technique, right? Tissue culture, egg inoculation, and we have a cell lines, we have a selective cell lines. Only if you provide this type of cultures for the cultivation of the growth of the virus, only then this can grow. But these viral particles usually will not grow in the artificial medium, as like in case of the bacteria and all these things. Na stands for nucleic acid. Okay, so separately we discuss about the cultivation of viruses, how the virus can be cultivated. The quite traditional technique is, we call it as a egg inoculation technique, egg inoculation. Then we have a culture technique, and then we have a cell lines. We have a HeLa cell lines. Even for coronavirus also, they're doing the cell lines actually. So we discuss separately when we talk about the cultivation. But things you understand, virus usually will not grow. Will not grow in what? Grow outside the cell. It means in the artificial in a medium, as like in case of other organisms. So this is one main feature. Another thing is it is an obligate parasite. It is obligate parasite. It means very strict in the requirement in the nutrition. For example, the particular virus which has the ability to grow only in a lungs means it grow only lungs. It grows in the WBC means grow only in WBC. Grow only the microglial cells means it grow only the microglial cells. So it's very selective in nature. So if the particular host cell die along with that host cell, the viral particles also will die. So this cannot switch over from one particular area, or this cannot change the mode of nutrient from one area to the other part, as in case of other organisms. So this is the one important feature that you could see in case of this case, like virus. So it is an obligate means obligate is a very strict parasite, and the selectivity and also the viral group virus are very selective in the mode of nutrition. So what type of cells it required? For example, HIV cells that you say it requires only in the WBC means it grow only in the WBC. Okay, so yeah, the same way, like it's very selective. This we call it as a cell tropism. This property is called it as cell tropism, where this particular, very much particular about the cell, the host cell. So almost it is like obligate. It means very strict parasite, and this also like unaffected. It's unaffected by antibiotics. Most of the times, like it may remain unaffected by means of antibiotics. Even give the bacterial the antibiotics also, like that cannot destroy mostly the viral groups. And most important thing is viral groups often undergoes mutation. This is very important. Even recently also, like you would have heard about, like coronavirus. There is other type of mutant coronavirus came. It seems to be highly contagious. So virus seems to be highly what? Highly have the ability to go for mutation. Okay, highly go for a mutation. So these are the main features when you talk about, and it's very specific in action. Very specific in action. So these are the important characteristics. Now we move on to the structure of a cell. So next we move on to the structure. And also we discuss about the size of the virus. Very shortly we discuss about the size of the virus. As far as structure is concerned, I'll show you the slides for the structure of virus. So we have the icosahedral, we have, here you could see. When you see the viral shapes are concerned, usually the viral shapes we classified. So how the viral shapes have been generally classified? We have three different shapes of the viral particles. We have icosahedral shape. The icosahedral shape, that is polyhedral. So this is polyhedral shape. I'll just mark it here. Polyhedral shape. So this is icosahedral. Icosahedral shape, and this is the complex. There are certain groups of viruses which seems to be complex here, complex structure, right? And you could see certain viruses which seems to be like helical, right? Helical, or it may be cylindrical also. It is cylindrical, and certain viruses are spherical in shape. Say like in case of like influenza, you take in a coronavirus. Coronavirus and all, it's like crown-like structure. You could see the crown-like structure in case of it's also like spherical, but the corona is actually the crown-like surface. You could see complex structures. You could see like a T for bacteriophage, mostly the bacteriophage which comes under this category. Coming to this polyhedral, how many phases you could see? You could see the phases, right? This polyhedral, like this. How many phases? Can you tell me how many phases that you could see like this? The polyhedral, there are like 20 phases that you could see. Example you take in case of adenovirus, polyhedral, the best example that you go with the adenovirus, poliovirus, herpes simplex virus. For icosahedral adenovirus, you take poliovirus. Then we have HSV virus. HSV stands for herpes simplex virus. Herpes simplex virus, poliovirus, adenovirus. All these viruses which comes under the category of icosahedral or polyhedral shape. How many phases that you could see in the structure? You could see 20 phases altogether. Okay, so this is a kind of virus which is got, and how you could see the symmetry. This is based on the symmetry, this symmetrical shapes and everything. How you can identify? You can identify by means of electron microscope, and the viral groups, viral particles, usually you could see under the electron microscope, not by means of the normal microscope. Next, we'll discuss about the size of the virus. We'll discuss about that. Fine. So, icosahedral poliovirus, and we have a herpes simplex virus, and we have the 20 phases in this side. Helical. Helical is a cylindrical. Example, you take tobacco mosaic virus. Example is tobacco mosaic virus, and we have influenza virus. We have influenza virus. This comes under the helical structure. Examples are tobacco mosaic virus and influenza virus. Then come to this part. This type of complex shape, neither icosahedral nor helical, but this is having something called as tail or comb-like structure. They have a tail or comb-like structure. Example for this complex structure, we have the T4, T4 vaccinia virus. T4 also you can take this virus is vaccinia, vaccinia, vaccinia virus. This comes under the complex. So based on the symmetry, this classified into polyhedral, helical shape, and the complex shape. Fine. Coming to the size and the shape of the organisms. Can someone tell me which is the largest virus? Size. Coming to the size of the viral groups we talk about. Okay, so retrovirus which comes in like spherical shape, retrovirus like HIV which comes under the spherical shape. Okay, so size is concerned, pox seems to be the largest virus, which is a 450 nanometer long. 450 nanometer long. This is the pox virus, which seems to be the largest one. Okay, 450 nanometer. But it is similar to, this size is similar to that of the bacteria. For example, you take a mycoplasma, rickettsia, chlamydia. These are like the smallest bacteria. So this size is equivalent to that of the smallest bacteria. So the largest virus itself is equivalent to the smallest bacteria. It means the pox virus you could see 450 nanometer long. And parvovirus is 20 nanometer. Parvovirus is the smallest virus you can take. Parvovirus, it's a 20 nanometer. The largest virus you take, the animal virus, it's a 450 nanometer. The pox virus, smallest virus is the parvovirus. It's 20 nanometer in length we talk about. But when we specifically talk about the plants, if you want to know, sir, I want to know about the plant, which is the smallest plant virus, or which one is the largest plant virus. Coming to the smallest plant virus, can someone give me example for the smallest plant virus, small plant virus, and the large plant virus? The small plant virus, if you take, it's a satellite virus. The satellite virus you take, tobacco necrosis virus. Tobacco necrosis, we call it as TNV, satellite virus, or we call it as tobacco necrosis virus, which is the smallest plant virus, which is of 17 nanometer only. This satellite nanovirus or the tobacco necrosis virus, which seems to be confirmed to be the smallest of all the plant viruses, right? So this tobacco necrosis, that is satellite tobacco necrosis virus of 70 nanometer, and the large plant virus is citrus, citrus tristosa virus. It's citrus citrosa is the largest of the plant virus, which is the largest of the plant virus. Citrus citrosa ristosa, I think. So I'm not sure about this. Ristosa, I think. So I write ristosa. You just confirm, guys, the spelling. Citrus ristosa virus, which confirmed to be the largest virus. Yeah. Okay. This is the size of 2000 into 12 nanometer. So this is of size of 2000 into 12 nanometer. This is the size for the plant is concerned, more specifically. Coming to the animal, which is the smallest virus. When you talk about the animal more specifically, animal, you take the small virus and you take the large virus. Can you give an example for smallest animal virus and which is the large animal virus? Coming to the small animal virus, it's a foot mouth virus, FMV, foot and mouth, FMV, foot and mouth virus, we call it as. So this is smallest virus, which is of 20 nanometer, just a 20 nanometer. But the largest animal virus is variola virus. Variola virus. And the variola virus you could see is a 350, 250 into 100. 350 into 250 into 100 nanometer. So this is the size of 350 into 250 into 100. This is the variola. But foot and mouth virus is only the 20 nanometer. This is the smallest animal virus. So as far as size is concerned, size, you could see the small, I given a explanation for the plant as well as for the animal, which is the smallest and which is the largest. In this part also you can get a question, which is the smallest animal virus or which is the smallest plant virus or which is the largest. If any part the questions comes also, I can now tell me like what might be the range. In general, if the question comes, what is the range of the size of the virus? I given like individual virus, the size and everything. In general, what will be the size of the viral particles in general, leaving all the exceptions. Like, if you talk, if the question comes, what is the general range of the viral groups? If the question comes, like, what will be your answer? Because it starts with 20 nanometers. From 20 nanometer to what will be the maximum size? That is actually the range of the viral particles. So that type of question also it will come. Okay, right. Yes, it's a cuboid. It's a cuboid. Maybe it is enveloped or it is a naked virus. It is polyhedral or it is a cuboid structure. So this is about the size of the virus. Now we move on to the classification. So classification of virus, as far as the classification is concerned, who proposed classification? Can someone tell me who proposed the who gave the classification of virus? Who gave the classification of virus? I'll go to the classification part. The classification. Okay. As far as classification of virus is concerned, classification of virus. This classification of virus, which was given by, which is given by Baltimore. Baltimore, we call it as a Baltimore classification. Baltimore classification. Baltimore classification. Can you tell me the year where he given this classification? I think it's 1971. Will you please confirm? Baltimore in the year 1971, he given the classification, and there is a council. This council call it as ICTV. ICTV, International Council of, what this actually ICTV stands for. ICTV, 1971, Baltimore given the classification of viruses. He made actually the classes of like 7 classes. We have the class 1, I think it's not visible. There are 7 classes of classification. He named as 7 classes of classification. The Baltimore classification is concerned. Okay. So this council for the viral classification, this ICTV, they classified into the 7 classes. If you could see the 7 classes of the classification which was proposed by Baltimore, you could see mainly the type of what are the main things that you could see. The virus seems to be the genetic material as a DNA, or the viral it seems to be a RNA. Or what will be happening, it may be a DNA is double stranded DNA, or it may be a single stranded DNA. If RNA, you take RNA is also it's a double stranded RNA, or it may be a single stranded RNA. This is one type of thing. As the International Committee of Taxonomy of Virus, exactly ICTV is the International Committee on Taxonomy of Virus. Again, all the 7 classes being there are altogether 7 classes. We have RNA viruses, certain viral virus or DNA, right? Variola is the largest virus. Okay, so DNA and the RNA. So it may be double stranded DNA, single stranded, double stranded RNA, as well as single stranded RNA. When come to the RNA, it may be having the positive strand, or it may be negative strand. Can someone tell me what is this actually positive strand RNA, or what is actually the negative strand RNA? Sometimes you would have seen the classes of virus. It's a positive strand RNA. Usually we give for a RNA is positive strand or the negative strand. So if you make it the stand for double stranded, it's like positive strand, or it may be negative strand RNA. What makes the difference? The positive strand RNA or the negative strand RNA? What makes the difference? What we actually mean by the positive strand sense RNA? What I mean by the negative sense RNA? So actually, the positive sense RNA, which will stand sense RNA, which will be similar to what? Similar to mRNA. It will be similar to. It will be similar to what? Similar to mRNA, but negative sense RNA. This negative sense RNA, which is complementary to mRNA. This usually complementary. This makes a difference. Complementary to mRNA. So this makes a difference. If someone that they mention as is a positive sense RNA, means this RNA is exactly the same like that of a mRNA, messenger RNA. If they mention as a negative sense RNA, negative sense means it's complementary to mRNA. If it seems to be complementary to mRNA, then what will happen? This complementary mRNA molecules will be complement. Then what will happen? Double strand RNA will be formed. Then what should happen before this negative strand, right? There are negative RNAs that has to be first converted to what? Has to be converted to positive RNA. Only then it will be going for the further reactions. Okay. So This is how actually the Baltimore classification. So one thing that you should understand, classification, who gave the classification? Baltimore in the year 1971. There are how many classes we have? There are 7 classes. What are the 7 classes that you could see? Right? 7 classes is like DNA based on what? Based on the genetic material, the DNA or RNA, whether it's single standard or double standard, whether it's a positive sense or a negative strand, or this also includes like its envelope. Certain viruses which have the outermost coat like envelope, and some viral groups are naked actually, or sometimes it is naked. So these are the parameters that we took as far as the classification is concerned, right? Sometimes what happened, you could see inside you have the capsid. The capsid is covered by the outermost envelope that you could see outside. Sometimes what happened, you could see only the naked virus, you could see only the icosahedral structure that you could see, icosahedral structure that you could see here. Only the structure of this is not covered by the outermost envelope. This seems to be the naked virus. This is a naked virus. And some virus, what happens like this is the inner part which will be having the icosahedral structures which covers by means of the outermost covering called as envelope. Okay, so this is how the classification has been made by ICTB by Baltimore, and there are how many classes they ask is actually there are 7. Coronavirus which comes under which class out of this? If the question comes like this comes under the 4, that is a 4th class, coronavirus which comes under the class 4 in the classification, the Baltimore classification, because we're talking much about the corona here. Corona comes under the 4th group of this classification. The reason why we're talking about the 4th group is because the 4th group of the classification, usually it is enveloped virus, or the enveloped virus which comes under the 4th group, and it is positive sense RNA. It is my positive sense RNA and is a single standard. So these type of virus usually which comes under the 4th group. 4th group, it means the coronavirus also having the corona is the enveloped virus and it's having the positive sense RNA and it is a single standard. So these are the features that you could see as far as corona is concerned. So if it is a positive, I told you like positive, it's similar to that of mRNA, similar to that of mRNA. So what is the weight of the coronavirus? How many kilo base pairs that you could see in coronavirus? There are 30 kilo base pairs. There are 30. Corona is concerned. This consists of 30 kilo base pairs. Out of 30 kilo base pairs, there are like 20 kilo base pairs or seems to be non-functional genes. Only the remaining 10 which seems to be the functional gene. That is what the actually the discovery shows. Out of 30, 20 seems to be non-functional. The remaining 10 kilo base pairs only seems to be the functional. Okay, fine. Here, you take this is the envelope virus, and this is a positive, which has got the positive RNA, and this is a single standard, which is a single standard. Okay. So this is about the classification of the viruses. So I mean, you need to understand the negative and the positive sense. Whereas the negative sense, so what has to be done? The negative, because negative sense is complementary to mRNA, so it has to be converted to the positive sense RNA by which enzyme? By usually RNA polymerase enzyme. RNA polymerase enzyme. Before translation, this process has to be completed. So this is about the classification of this viruses. And one more thing, when you talk about this virus, the family, the coronavirus, which family it comes under, the corona which comes under the family called as nidovirales. The family we're talking about corona is nidovirales. Nidovirales is actually a type of family, that family where the coronavirus which falls under nidovirales, and this lacks actually exoribose nuclease activity. Corona also having like exoribonuclease activity, and this is to what? Exoribose is to what? Is to limit to infect the host cells. So very detail even discuss about the replication, the life cycle of corona also. Fine. We done with the classification, and then we move on to the nomenclature. Nomenclature we call it as cryptograms. What do you mean by cryptogram? We talk about cryptogram, cryptograms, that is nomenclature of virus. Usually how this nomenclature of virus can be given. This was first proposed nomenclature of cryptogram of viral particles is given by Gibbs and Harrison. This was given by Gibbs and Harrison. This nomenclature has been given. As far as this part is concerned, so what are this consists of 4 pairs actually. When we give for a nomenclature for viruses, this consists of 4 pairs. For example, you take, I give an example for tobacco mosaic virus. Just one example I'll give for tobacco mosaic virus. This can be represented as R125 and EEN. We make it as yes, yes. So we make it as yes, yes. We have these four pairs that we could see in usually like when you go for the cryptogram or the nomenclature of the virus, which is proposed by Gibson Harrison. What is this first pair? The first pair is actually we call it as a nucleic acid. Based on the nucleic acid, this can be given. Nucleic acid, whether it is RNA or it is a DNA. So based on this, it's a coronaviridae family, but this, even the coronaviridae, when we talk about this, comes under nidovirales. The nidovirales is a very common under the SARS virus, under which when you go even detail, subgroups of subfamily, we go only we could see coronaviridae. But in general, the common family we talk about corona is a nidovirales. Okay, okay, fine. Now, so first is the nucleic acid based on the DNA and RNA. It's a single strand, double stranded. This first pair has been given. And second pair you could see is a molecular weight. So the second pair is for the molecular weight. For example, 2 bar 5 here you could see 2 is for about 2 million base pairs and 5 is the percentage, the total percentage that you could see 2 bar 5. And E bar E, this is the shape actually. You could see the shape of the virus. Maybe it's elongated or helical. Based on that, they're given the letter, the shape. The third pair is for the shape, and fourth is actually the host infection. It is for the host infection. So this is just one example how the nomenclature usually we give. For example, you take the tobacco mosaic virus. Tobacco mosaic virus, based on its first pair is for nucleic acid, second pair is for molecular weight, and third pair is for the shape, and fourth is for the host infection. For example, host infection means SS means, for example, you could see usually it affects as for seeded plants. Usually it affects the seeded plant and it affects the sap. So that is actually the first word they given for that as a clue, as a hint they given. So these four, the pairs that usually we give as far as nomenclature of virus. This we call it as cryptograms. This proposed by Gibbs and Harrison in the year 1968. 1968. This nomenclature of naming of grouping of the viral particles have been done. So we're done with the nomenclature. Next, we move on to the description of some important viral groups. Now we move on to the virus. We talk about the plant virus as well as the animal virus. Coming to the plant and animal virus, first we'll start with the plant virus. Coming to plant, yes. So common diseases that you could see in the plants. Your tobacco mosaic virus, potato leaf curl virus, and we have the tomato leaf curl. The common viruses that you could see: tobacco mosaic, potato leaf curl viruses, and tomato leaf curl viruses. We call it as plant viruses, that is TMV, rod shape viruses, that is RNA, and we have the size tobacco mosaic virus. When you talk about the size of the tobacco mosaic virus, it is 3000 angstrom. Size is 3000 angstrom by 170. The size of the tobacco mosaic virus, and this was first identified by Ham Frankel. And they first identified the tobacco mosaic virus, which consists of RNA, and the capsiomeres. So the capsiomeres, how many numbers of what's the number of capsiomeres that you could see in the tobacco? I mean TMV capsiomeres. Capsomeres are concerned. The total number of capsomeres in this case, it's 2130 in number. So you could see 2130 capsomeres which present in the tobacco mosaic virus, in which the nucleocapsid is 17 nanometer. The size I'm talking about, the nucleocapsid. The nucleocapsid is 17 nanometer. Sometimes the tobacco mosaic virus, any questions come in depth, the capsomeres, how many capsomeres that you could see in numbers, that is 2130, and nucleocapsid is 17 nanometer. This is about the most important plant virus issue that you could see in the textbooks that they would have given. Okay, so we're done with the virus. Next, we move on to the viral diseases of the plants. When you talk about the viral diseases of plants, which virus is responsible for rosette? We have the rosette disease, right? Can someone tell me which plant this disease which causes rosette disease? Tobacco mosaic virus for tobacco plants, right? Yeah. Yeah, you said like mimi virus. Mimi virus is of course like it's the largest plant virus which has been as if it's counted, but it's not been published legally. Like most of textbooks you could not see the mimi virus. Mimi virus which has not been published, most of the textbooks you could not see. Okay. But if you could see search in Google and other places, like you could see the Mimi virus, which seems to be the largest parasites, but it's not been given like officially and mostly in the textbooks. Okay. Rice ingro clans is caused which so you could say rice ingro viruses, which group of viruses it comes, like rice ingro disease, which caused by which type of viruses? Ratish, you asked rice ingro disease, which type of viruses usually this disease which caused by. Okay. Mima, I told you asked like Mimi virus is human virus. Yes, but it's because of the mutation. There is not like plant viruses or animal viruses. I told you already. Viruses are subjected to high rate of mutation. It depends on the mutation rate. If the mutation seems to be high, it's not to be very selective. Only it's going to affect only the bats or only with the particular animals. This can also affect the human beings. This all depends upon the mutation. So there's no exception. We cannot think like mini virus cannot infect the or affect the human beings. This can also affect the humans. Okay. Rosette. Rosette usually which usually cause disease in a groundnut. Usually this cause disease in a groundnut. Coming to the animal viruses. How do we categorize animal viruses? Okay. So Rosette disease which caused in a groundnut. Groundnut. Coming to the animal diseases, the common cold virus, the normal common cold virus which caused by rhinovirus, smallpox which is completely eradicated, so pox virus, rabies virus, infectious hepatitis, we call it as hepatitis of many types like HBV, HAV, we have different hepatitis, HAV we call it as hepatitis, HCV also we have, right? But mostly hepatitis B virus is a bloodborne, so hepatitis virus which cause measles virus, polio virus, polio, and influenza virus, encephalitis, and we have the mumps. In general, the viral diseases can be classified into dermatophytic, viscero, and the neuro. So how the virus can be classified? The animal viruses can be classified into dermato. Mostly it affects the skin region, the manifestation that you could see the skin. Viscero. Viscero means the viral particles usually which affects the what is it called inner parts of the body. And we have neuro. We have the neuro type of viruses. So we have three types of animal viruses based on the clinical manifestations. The dermatotrophic virus. Can someone give me an example for dermatotrophic virus? Measles, mumps. These are all like viruses. Yeah, which comes under dermatotrophic. Viscero type of viruses. Viscero type of virus you could see jaundice you take. Jaundice is a classical example because it affects inside internal organs. The neuro example you take polio virus. Polio which affects the neuro cells. So, based on the clinical manifestation, the virus can be classified into three types: dermatotrophic, viscerotrophic, and neurotrophic. Okay, these are the three types of viruses. Next we'll talk about the next part is replication. As far as replication, before going with the replication, let me show one more slide so that you can get detailed idea for yeah, I think this is not visible. Guys, I'll just dictate it for you. There are other type of diseases. Guys, it's not visible. I'll agree. I'll just dictate. These are the diseases that you could see. What are the diseases here? AIDS. AIDS is actually the viral disease. Chickenpox, Ebola virus. So, a couple of years back, we have called it as like the Ebola virus, hepatitis B virus, herpes virus. We have the herpes virus, influenza virus, measles and mumps virus, even virus which cause influenza virus which cause pneumonia, polio virus, rabies virus that is caused by rhabdovirus, smallpox virus, smallpox is a disease which caused by variola virus, but this was completely eradicated. Okay, so then yellow fever is caused by flavivirus. Usually this caused by the mosquitoes by means of mosquitoes, the human and the two plays a host. We could see the completion of life cycle, human as well as in mosquitoes. And SARS virus, SARS expansion, yeah, SARS expansion, severe acute respiratory syndrome. Zika virus, usually this comes under the what is it called, the rodents and a type of insects borne type of like agents usually which is responsible where you can sand wills and the staples sand houses where you could see that type of areas where by means of particular type of insects which has got the responsibility of carrying this type of diseases by, that is Zika virus. And SARS, severe acute respiratory syndrome, and we have FMD. FMD is foot and mouth disease. Foot and mouth disease, and the pathogen for the foot and mouth disease is Afte epizooticae. FMD is caused by Afte epizooticae. Here you could see the reservoir for a foot and mouth disease is domestic animals. Like if you have close proximity with, you see like cattle, goat, pig, sheep, right? Even from these reservoirs also you can get this foot and mouth diseases. Okay, so yellow fever initially with the mosquitoes and the human, and smallpox formerly it was with the humans. Since it has been eradicated, now it exists only in the research labs. Like many has been eliminated. Many has been eliminated. Okay, rabies virus you could see usually in the domestic canids, like you could see the dogs, foxes, in these cases, bats and raccoons, you could see the rabies, and usually rabies by you could see through the dogs it can spread. Pneumonia, human, measles and mumps. Usually that's the reason why we give the vaccine, right? MMR vaccine. MMR vaccine that we give for this measles and mumps and rubella. Measles, mumps and rubella. For this we give the vaccine, MMR vaccine. This for the human beings. And herpes virus is again for human. The virus is herpes simplex virus. This is responsible for shingles. In shingles, mostly in the immunocompromised individuals and all, you could see the shingle kind of like lesion, that formation which takes place usually by means of herpes virus. Hepatitis virus is bloodborne, where this cause infects only the human. Ebola virus, it's especially they ask which type of virus, which pathogen is responsible for Ebola. Filovirus, which is responsible for Ebola. Chickenpox, we have human herpes virus, that is varicella zoster, we call it as, and AIDS, which is caused by HIV, that is acquired immunodeficiency syndrome. So these are the list of diseases. I hope this font is not clear for you. I'll send this image to your group. Okay, since I took from the textbook directly, reference book, so I couldn't increase the size. So sorry for the inconvenience, guys. I'll send this picture in the group. Okay, these are the some of the common animal diseases and the pathogens and the reservoir this particular thing shows. Okay, next we move on to the, as I told you, the animal viruses which classified into dermatotrophic, viscerotrophic and the neurotrophic. Coming to the next part, we will discuss about what we call it as retrovirus. This is one type because it's given in the textbook also, in the 12th textbook NCERT also you could see this type of virus. Okay, retrovirus. Coming to this retrovirus, the retrovirus is actually, yeah, the retrovirus, it's actually what is a genome that you could, genetic material that you could see in case of retrovirus. What is the genetic? It's RNA or DNA. Retrovirus is a single stranded or double stranded, right? So first we'll start with the retrovirus. Here, retrovirus is a single stranded RNA virus. Single stranded RNA virus, and this consists of mainly 8 important protein, and this is most mutable virus. Most mutable virus, the retrovirus, and this particular virus which causes retrovirus that we're going to discuss about HIV/AIDS, that is human immunodeficiency virus. This cause acquired immunodeficiency syndrome. So what we call it as syndrome here. So syndrome. Why we use the word called a syndrome? What do you mean by syndrome? Here it's not a disease, it acquired immunodeficiency syndrome. Why we given the particular term called a syndrome? What does a syndrome means? We call it Down syndrome, Klinefelter syndrome. Why we given for this particular disease as a syndrome? Is there any justification why we call HIV, the AIDS, as a syndrome? Okay, so when we look at the structure, of the HIV virus. Coming to the structure of HIV virus, this you have seen already in the textbooks. So this is the structure, the spherical shape that you could see. Okay, mostly mostly mutated virus, because it's one of the main feature of the viral groups actually. But in that, retrovirus is highly mostly mutated virus. It keeps on changing its structure, that's the reason, mostly mutated virus, where we cannot discover vaccines or any type of drugs, because today it might be in one particular shape. Right? If we design a vaccine or any drug for that particular viral groups based on the receptors, tomorrow it may again change its shape actually. This we call a genetic drift. That is antigenic drift, we call it as, right? This keep on changing its structure. It will be keep on mutated, like it will be keep on changing its structure as well, right? So coming to the HIV, this is the HIV structure, human immunodeficiency virus structure, and many people who first identified HIV virus, because I told you in the historical perspective who first discovered the HIV virus. This already I discussed. Coming to this part, here we have the glycoproteins. What are the different types of the glycoproteins we could see? This GP120, the outermost the protein you could see, GP120, GP41. We have GP41, and inside we could see GP9 also. GP120, GP41, and we have like P17. Inside we could see GP9. Right? These are the different glycoproteins, and these glycoproteins are playing a very important role in diagnostic procedures where you could see usually you perform the test called ELISA, you perform TRIDOT technique, you perform Western blotting technique, where you could see these glycoproteins, which plays a very important role due to the presence of these glycoproteins in the serum sample. Based on this only, you categorize ELISA as a direct ELISA, indirect ELISA, or we call it a sandwich ELISA. We have different types of ELISA, the techniques we have. How these techniques are applied, right? This based on the presence of what? Presence of these glycoproteins. These glycoproteins are part of the virus where you could see the presence of the glycoproteins in the serum sample by which we raised antibody against this particular proteins and we try to confirm the patient is positive or negative. We look for the antigen itself, then we call it as a direct ELISA. We look for an antibody in the patient serum, then it is an indirect ELISA. There are two types of ELISA. Usually we call it a direct ELISA and indirect ELISA. Direct ELISA in the patient serum, we could see the presence of the antigen itself, the glycoprotein itself, then it is a direct ELISA. Where we look for the presence of antibody, then we call it as an indirect ELISA. So by means of this, we call it as direct and indirect. So this is the significance of this glycoproteins which present. This is like in a fusion proteins we could see. These are the fusion protein, the green part that you could see, right? These are the fusion proteins we call it as. And not only that, this glycoproteins also play a significant role in the designing a drug. As of now, you know pretty well, there are no proper vaccine, no vaccines for HIV virus, but we have drugs to stop, not to eradicate, not to kill the HIV virus. As of now, we have the drugs to just stop the replication of the viral particles. For example, you take as this particular drugs that we gave, what this drug will do is this will stop the replication. This will stop the replication of the viral groups, but it not destroy, not kill GB120, or it may block GB9, that is your reverse transcriptase part. So somewhere the drugs which is got designed, right, this go and block the particular area of the virus so that the replication of the virus can be stopped. And also the problem for designing the drugs for HIV virus is like it's keep on changing its shape. It's highly mutated virus, so it keeps changing its structure. So it's very difficult to find a proper vaccine or proper antibiotics for this one. But as of now, we have the drugs like azithromycin, and this just stop the replication of the virus. Okay. So what are the structures that you could see? This enveloped virus, of course, this consists of RNA. You could see the RNA inside, and we have the capsid. This you could see the capsid part. This is a capsid, and this capsid where inside you could see the but main enzyme which is there inside, which we call it as a reverse transcriptase enzyme. As I told you already, the reverse transcriptase enzyme, which plays important role in the reverse transcription, where the DNA is usually in the central dogma, DNA converts to the daughter DNA, the parental DNA which converted to parental DNA converted to daughter DNA which gets converted to RNA. That is the RNA, then RNA which will be eventually converted to the protein. This is what the normal process which takes place. But what happens in this case, as far as reverse transcriptase is concerned, where the RNA which will be converted back to a DNA. This by means of an enzyme called reverse transcriptase. Who's performing this process? Reverse process by means of the enzyme called reverse transcriptase enzyme. This process called as reverse transcription. So reverse transcription. For this only, the person I told you, like they got the Nobel Prize for identifying this process. Feminism we call it as. Feminism is a process where they identify the reverse transcription enzyme by means of the enzyme called reverse transcriptase, where the RNA is getting bounced back and getting converted to the DNA. So why it has to be converted to DNA? Because as we discussed, this is a RNA virus, whereas it's going to affect the WBC, right? The WBC is what? It's a DNA virus. It's a DNA virus. So what happens, for example, this is a WBC we're talking about. This is a WBC. WBC which consists of double stranded. This is a DNA. For example, this is your HIV virus you're talking about. The HIV virus is a single stranded RNA virus. This is RNA virus. So if at all this virus, the genetic material has to go and integrate with the DNA. This is a single stranded. Okay, this is a single stranded RNA, and this is a double stranded where it has to be converted. So for this conversion, because this has to go and integrate with the host DNA, for that it requires a process called as reverse transcription. The reverse transcriptase which perform the function where the RNA will be getting converted to DNA, then the converted DNA will go and integrate with the host DNA, right? Okay, for this is the main reason why the reverse transcriptase is phenomenon mechanism which is happening in the HIV virus. This is the integrating pathway where lysogenic cycle takes, because I told you there are two types of life cycle, lytic cycle and lysogenic cycle, where the HIV virus often takes the lysogenic cycle. Lysogenic cycle. Okay. So this is structure of HIV, the virus. Next, move on to the life cycle. Coming to the life cycle of this virus, life cycle of coming to life cycle. Here you could see this would have seen in the textbook itself. Okay. Yeah, RT. We come to that. RT-PCR is used to detect for corona. Yes, because it's for RNA. RNA. Okay, RNA. But real normal PCR is for the DNA. Real-time PCR is for RNA. That used for the corona. Okay. Then here you could see just a quick outline that I'll give how this occurs. The replication of retrovirus. This HIV virus where the virus having the RNA core and this is getting infected with the host. This is the animal cell we're talking about, which get adhere under the plasma membrane of the animal cell. And the viral RNA is introduced into the not entire cell, only the genetic material which is going inside. Now what happens here? The viral RNA is getting converted to DNA. I told you the RNA is getting converted to DNA by means of the enzyme that we call it as a reverse transcriptase enzyme. Now what happened? This viral DNA integrates. I told you why it has to be integrated, because here it is a DNA, but here this is the RNA. The RNA has to be converted to DNA because it's going to integrate. For that, it requires an enzyme called as reverse transcriptase enzyme, where the RNA has been converted to DNA. Now, what will be the fate of this DNA? Now, this DNA incorporates or integrated with the host genome. So, when the host DNA starts replicating, the viral particles which has got like integrated, that also start replicating. This also start replicating, and now the new viruses are produced. Then this like takes a lysogenic type of life cycle where it's not destroying the viral cell, where new progenies or new number of cells which keeps on increasing, the new virus can infect the other cells. So now what happens in this case, the viral load which will be keep on increasing. So it means these cells we're talking about, right? These are actually all the WBC cells. The white blood cells are all infected with what? Affected with the HIV virus, which is carrying the virus itself inside. So you would have studied once the viral load started increasing, there will be the reduction in the CD4 count. Can someone tell me what is the CD4 count? As far as like usually in case of HIV patients, like where you could see the viral load will start increase because the numbers which keeps on increasing, all the fresh cells which is coming out of the replications, all the new cells, the WBC cells are getting affected with the HIV. So the viral load is increasing. The viral load is increasing. Same way where the CD4, what is CD4? We call it as this CD stands for cluster of differentiation. We have two types of cells, CD4 cells and CD8 cells, which performs immune functions in our body. In particular, where the person has got affected with HIV, where the CD4 level counts will start reducing. When the CD4 level start counts reducing, what happens? The T helper lymphocytes function also will start reducing. Then this point of time, what happens? When the CD4 will start declining, then the person will become immunocompromised or immunosuppressed. So immunocompromised or compromised or immunosuppressed. And you should understand it's a collection of disease. So that's the reason we call it as a syndrome, where the HIV individuals are not getting killed because of only the HIV virus. The person is getting killed because of the infections that we call it as opportunistic infection. So once the yeah, I'll give you a break, guys. I'll give you a break. Okay. So let me complete this one. Okay, just give me 2 minutes time. Okay. So how the patients are getting killed, HIV individuals are getting killed, this is not because it's not so readily. Of course, HIV can conquer the world and everything. But thing is, how the person is getting killed. This is because of the opportunistic infection. Why? Because the CD4 counts goes down. The CD4 counts goes down. The T helper macrophages, this also goes down. Then automatically what will happen? The person will become suppressed immunologically. He or she will become suppressed, and he or she will become exposed to what? Opportunistic infections. Opportunistic infection, for example, you take tuberculosis, cryptococcosis, for example, skin cancers like Kaposi's sarcoma. These are the different diseases where because the person is giving opportunity for the this type of infections. So the persons are getting killed because of the opportunistic infection, not just only because of the HIV virus, because only function of HIV virus is to reduce the CD4 count. Thereby, what happens, like it suppresses the immune system. So all other opportunistic infection gain entry inside. So time is now 10:53, guys. So we go for 5 minutes break. Once we complete with the break, like we'll start with the next part. Okay, just 5 minutes, guys. Come back soon. Welcome back, guys. So after the break, fine. So we are discussing about the replication of the HIV virus. So the other things that you should remember when talk about the HIV virus is what type of replication the cycle is. This is called a spinocytic replication. So often they ask what type of replication the HIV virus which takes place. Which takes is a pinocytic. Pinocytic is a type of replication, and with the gene where you could see the RNA is getting converted to DNA, and the enzyme by means of like RNA dependent DNA polymerase. RNA dependent, this by means of RNA dependent DNA polymerase. So this is the end part, right, of this HIV life cycle. Fine. When come to the next part of this, the virus is the virus and the cancer. There's a link between the cancer and the virus. Can you tell me the genes which are responsible for causing cancer? The genes which are responsible for causing cancer. In this case, virus and cancer. The genes are called oncogenes. These oncogenes are responsible for causing cancer. And usually you know to categorize this cancer or the tumor. The cancer can be classified into benign tumor and the benign. We're not just discussing about the tumor here. We're discussing about the virus and cancer here. There are certain viruses which are responsible for causing the cancer as well. You know the difference between the benign tumor and the malignant tumor, which is harmless here. Benign is harmless, which is harmless, and malignant tumor which will be giving this deadly effects to the person. The reason is because in this case a benign tumor where the tumor might be confined to one particular location, where in case of what happens a malignant tumor, it's deadly manifestation because the cancer tissues like which will be get disseminated through the bloodstream wherever the blood flow occurs, like even the other organs or other parts of the body can also be affected in case of malignant. So malignant is a deadly. So we're not talking about a cancer, but there are few viral groups which are evidenced in causing cancer. That means these viral groups are carrying oncogenes. Example for oncogenic viruses. Can you give examples? Like you take adenovirus, herpes virus, pox virus, virus. Then we have virus. And most commonly you could see like type of skin cancer. Skin cancer also it's quite common by means of, we call it as Kaposi's sarcoma. Kaposi's sarcoma. For attention guys, we're not talking anything about the cancer now. We're just relating the virus and the cancer. For that one I just took this part. Okay. So these are some of the virus which is inquiring with the oncogenes. Adenovirus, herpes virus, pox virus, 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 and here the Kaposi's sarcoma, which is a skin cancer. This is quite common in HIV individuals. They often get this type of cancer. HIV immunocompromised individual, they get this type of cancer. Okay. Out of this was first the virus and its cancer, the oncogenic property was first identified by William Ellerman. William Ellerman and Wolfgang. I'm not sure with the spelling, guys, for the Wolfgang, but William Ellerman and Wolfgang. These two guys are contributed. Wolfgang or Wolfgang, I'm not sure about this. So just kindly check. So William Ellerman and Wolfgang, they contributed the association between the cancer and the virus, and out of which all the total cancer in the global standards where the virus is responsible in causing 15%. 15% of the cancer usually which is caused by the viral groups only. In that way, like virus also having a significant role in causing the cancer because these viral particles do also carry genes. The genes are oncogenes. Next, we move on to the phage viruses, the bacteriophages that we'll discuss here. Coming to the bacteriophages and other phage varieties. So this is a typical structure of the bacteriophage we're talking about. So we now move on to the phage virus. What do you mean by phage? Phage means a virus which affects the other groups, maybe a bacteria, maybe actinomycetes, maybe yeast, or maybe a cyanobacteria, maybe anything, right? So if this affects bacteria, the virus which affects the bacteria, then we call it as a bacteriophage. The virus which affects actinomycetes, actinomycetes, what is that called? Then this can also affect uh, actinomycetes. If it affects a yeast cell, then cyanobacteria, then we have algae, then fungi, like mold especially. So it doesn't mean like the virus which affects only the bacteria. That we usually we call it as bacteriophage. The virus also has the ability to infect or affect the actinomycetes. This we call it as actinophage. This we call it as a actinophage. This is a symbol that usually we give for a phage, actinophage. And yeast is actually we call it as zymophage. Zymophage. So cyanophage, cyano, C-Y-A-N-O. Cyanophage is for the cyanobacteria. Algo, this we call it as a phycophage, P-H-Y-C-O, phycophage. And this is mycophage, which affects of fungi is called as mycophage. Okay, these are the different the phage viruses we're talking about. But in the textbook, often they given mostly the bacteriophage. This bacteriophage, which has the ability to affect the bacteria, and this takes two types of life cycle that I told you already is lytic cycle and lysogenic cycle. This take lytic or it take a lysogenic cycle. Mostly this viral particles, right? Yeah, cyanobacteria. It's a cyanophage. Cyanophage. It's a cyanophage. And this it takes lytic cycle or lysogenic. Which group of bacteria usually it prefers? It prefers coliforms. It prefers coliforms because those organisms which normally present in your intestinal part, the colon part, right? So usually that's why we call it as coliforms, right? Since these phages have the ability to affect this type of bacteria, right? No, the phages have the different structure. For example, you take phage X174. Phage X174 is having a different structure. Bacteriophage is having a different structure, but the complex structure, the tail and comb structure is there. The spike and everything will be there, but it depends upon the molecular weight and the size, the number of genes. This keeps like varying between one phage to the other, but all comes under the complex structure only, like comb or tail structures only, but slight differences that you could see among the phages. So we talk about bacteria, the bacteriophages. Usually the coliforms are the target of these viruses. Usually it affects. Coming to the virulent, the lytic phase, usually T2 and T4. T2 phage and T4 bacteriophage usually which goes with the lytic cycle. When we come to lysogenic, we have the lambda phage. We have the phage called as lambda phage. Sometimes they'll ask questions from this. Which among the following which takes the lytic cycle? Which of the following will takes the lysogenic cycle? Lytic usually which goes with T2 phage and the T4 phage. Lysogenic, if the options comes, it's like lysogenic means this will not kill the host cell. Where the lytic cycle, this will destroy the host cell, then offsprings, the new viral particles can be produced eventually after destroying the host cell. So lytic and lysogenic. And coming to this part, the structure of the bacteriophage. This is usually the bacteriophage which affects the E. coli. Which part you didn't understand, Kodai? Because we are in the phage viruses we are talking about. These phage viruses are the virus which affects different group of organisms. Say like if it affects bacteria, actinomycetes, yeast, cyanobacteria, algae, fungi. These are the different names that we give: bacteriophage, actinophage, zymophage, cyanophage, phycophage for algae, mycophage is for fungi. Right. So when we talk about the structure is concerned, when we talk about the structure, this bacteriophage structure will slightly keep varying. All the bacteriophages or all the phages will not be having the same structure, right? So it will not be having the same structure. It will be differ from one to the other, but all comes into the complex structure because we discussed lycocytoidal structure, we have the spherical structure, we are complex. Usually phages comes into the complex structure, but all structures may not look alike. This bacteriophage, especially which affects the coliforms, that is the bacteria which present in the colon part, where it takes two types of life cycle: lytic cycle and lysogenic cycle. Lytic cycle, what happens when the virus infects the bacteria or the particular host cell? What happens? This will destroy the host cell. At the cost of destruction, the new viral particles will be produced. This is lytic cycle. Definitely, destruction of the host cells takes place. Lysogenic cycle, no destruction of the host cells takes place, where the viral genetic material integrates with the the host DNA, and this will be the part of the host cell. It will be the part of the host cell, and this will become like which will be keep on multiplying inside. That is a lysogenic. So lytic is actually virulent. Lytic is a mostly the virulent because it's destroying the host cell, right? Why yeast and fungi is separated? Because as far as the fungi is concerned, fungi is in two form: yeast form and the mold form. This fungi is actually, this we call it as a mold. We're talking about the mold. I hope you remember fungi has two types: mold form and yeast form. So we categorize yeast as a unicellular and mold as a multicellular. On that basis, we classify. For example, you take Saccharomyces, these yeast variety, Candida, all these things that you take. But mold you take, Aspergillus, Penicillium, Rhizopus, Mucor, like all the things which comes under this part. So based on this, we classify yeast and the mold separately. Okay. So lytic is a virulent because it destroying the host, and this lysogenic can be also called as a virulent. Coming to the structure, as far as the bacteriophage structure is concerned, this is the head part. This is the head part you could see, which is a 1,000 angstrom ecosahedral structure, which consists of DNA inside, coil-like structure DNA inside the head, and this part, this is the caudal part. This part is the caudal. This caudal being connected with the tail. Again, it's a 1,000 angstrom here. And this is the base plate which carries the spikes. We have the three pairs of spikes that you could see, especially with the T2 fudge that you could see, right? As far as the double standard DNA, it's a double standard DNA that you could see, which is of 53 micrometer. This is a 53 micrometer. Size of the DNA that you could see is a 53 micrometer. Ecosahedral, how many capsomeres are present in the ecosahedral structure? It is 2000. 2000 capsomeres are present in the head region. 2000 capsomeres are present in the head region, and the DNA size is 53 micrometer. And the tail part that you could see, the tail which is comprises of 80 angstrom. The tail which consists of 80 angstrom in diameter. This is for the tail. The tail is 80 angstrom in diameter. And the rings that you could see, right? The rings, how many rings that you could see is at 24 rings. More specifically in T2 bacteriophage, if you could see the rings, right? The lines here. This you could see 24 rings that you could see. Exactly in T2 fudge, you could see 24 rings, and each 24 rings which consist of, each rings which have the 6 subunit. Totally, you have 144. How many subunits that you could see? 144 subunit. 144 subunits you could see. It means each it means 24 rings. The 24 rings meaning the 6 subunits in each. 6 subunits in each. So altogether you could see like 24 rings and 6 subunits in each. So altogether 144 units that you can see. So this is about the structure of the bacteriophage. Usually this affects the coliforms, the bacterial cell. So based on this, this classified into lytic cycle and lysogenic cycle. So this is a diagrammatic representation where you could see lytic and lysogenic. Coming to reproduction is concerned, the phage is concerned. I told you lytic cycle which goes with the T4. But lysogenic is usually the temperate. Lysogenic can be also called as temperate. There is other name for the lysogenic cycle is a temperate cycle or lysogenic cycle or avirulent cycle. Lytic cycle, this is a virulent cycle. This is a, since it's destroying the host cell, this is a virulent cycle. Lysogenic can be also called as a temperate cycle. This temperate cycle usually occurs in the bacteria, not only in bacteria. It does in bacteria, and also this is going to affect in the other groups. If this lysogenic cycle is going to affect the bacteria, right, and also this is going to like animals, like plant cells, you take any cells. If it is like the viral DNA getting integrated to this bacteria, then we call it as a prophage. The name is the prophage that we'll give. If the same viral DNA, if it's getting infected, the host is like other than the bacteria, then we call it as a provirus. So prophage that you give only for what? Only if the viral DNA gets integrated with the bacteria. If the host is a bacteria, then it is prophage. If the host is other than the bacterial cell, then integrated after integrations, we call that as a provirus. So this is about the life cycle. When you look at this diagram, where you could see the phage get attached to the host cells and injects its DNA. Injection of DNA takes place, and there is no connectivity between the host DNA and the viral DNA. You could see. Then what happened? It leading to a separate life cycles where the phage DNA circularizes and enters the lytic cycle. It may decide whether it go to the lytic cycle or it go to the lysogenic cycle. If it go to the lysogenic cycle, your viral DNA that will get integrate with what? Integrate with the host DNA. If it is a prophage, it's understood this must be the bacteria. So because I told you, if the host cell is a bacteria, we use the word called as prophage. If it is other than the bacteria, then we use the word called as provirus. It's a provirus. Now, what happens? This goes to the lysogenic cycle. As a part of lysogenic cycle, the lysogenic bacteria reproduces normally as the normal cells multiply. Your viral particles are also, the genetic material also will start multiplying inside. Then this will start keep on accumulating inside. Where the cell lysis, the lysis will not take place. Destruction of the host cells will not take place in this case. But the other hand, you can take lytic cycle, where this is actually is not the part of the host DNA. New fast DNA and proteins are synthesized, assembled into the parts called as virions. The virions are formed. In this case, we could see the virion structures getting formed, and here the cell lysis takes place and releasing the new infective agents that we call it as the virions. And now these virions which go and affects the other new viruses. Here the lysis occurs, the destruction of the host cell occurs. Here there is no destruction of the host cell. Okay, so thus we maintain the difference. At any point of time, this can, it doesn't mean like it can take only the lytic cycle or lysogenic cycle. So either lytic cycle or lysogenic cycle, or sometimes it take both. So the viral, the bacteriophages can take both lytic as well as lysogenic. It's not confined decision. It should go only with the lytic or lysogenic, or it can go with the both, right? So it's not adamancy or specificity. It should go only with one type of cycle. So this is about the phage viruses. Coming to the subviral agents. The subviral agent, talk about subviral agents. Subviral agents. As far as subviral agents are concerned, okay, subviral agents are concerned, you could see what are the things: satellite virus, viroids, and virusoids. The three things that you just cross check whether you are aware of all this term. Satellite virus, viroids, viroids, virusoids, virusoids, and also we have something called as prions. These are other areas, the subviral agents also you'll be discussing about, because we have only like another 15 minutes of time. I try to just give an outline only. So subviral agents, satellite viruses. This satellite virus, which was first proposed by Kursanin, in the year 1966. Viroids, which was given by Theo Diner in the year 1971. Coming to the satellite virus, this is a small RNA molecule. This is actually like same like of the virus. This is a small RNA molecules. We call it as a satellite viruses. This depends on what the satellite. This depends on another virus for the multiplication. This cannot function of its own. This depends on what? This depends on the other satellite virus or the viruses. This depends on other viruses for the multiplication. Example, you take hyperparasites of viruses. This depends on other virus itself. Example, you take hyperparasites of virus. So satellite virus are small RNA molecules which depends on the other viruses for the multiplication. Example, you could say helper luteovirus, nipovirus, helper sobinovirus, then helper dependovirus. These are the type of virus that you could see which comes under the satellite viruses. It means it depends on the other virus for multiplication. Coming to the viroid groups. Viroid, it's a small circular single stranded RNA. This is small single stranded RNA molecule. Simply we call it as a viroids. Viroids is actually which was in, it differs from species to species actually. For example, you take potato spindle where the number is 359 nucleotides, where you could see in a citrus exocortis you could see 375 nucleotides. So why I'm telling all these things, these are the subheadings where you discuss in subviral agents. For that you should understand what is viroids, what is satellite virus. And coming to the virusoids, as far as virusoids are concerned, what we discuss virusoids are actually described by J.W. Landl. This proposed by J.W. Landl, the person who first discovered virusoids, J.W. Landl. Okay, in the year 1981. This is also similar to that of viroids, but located inside the protein coat of the true virus. It's present inside the protein coat. Where it is present? It's present inside the protein coat of the other virus. Protein coat of other virus. It's present already. It's present inside. So virusoids proposed by J.W. Landale, which present inside the protein code of the other viruses, and this is necessary for the replication of the larger RNA. It will be facilitating the true the viral particles. Prions, prions, it's absolutely no nucleic acids. No nucleic acid, guys. But here you could see only the protein. Na is nucleic acid. Prions, which doesn't have the nucleic acid. In prions, you need to understand like this is responsible for neurodegenerative disease. Prions, you need to know the examples for the neurodegenerative disease. Prions is responsible for neurodegenerative disease. Can you give some example for this disease? Here you could see mad cow disease. We call it mad cow disease, especially in case of cattle, or Creutzfeldt-Jakob syndrome. CJS we call it as CJS is a Creutzfeldt. Often they ask a question: which among the following is responsible for caused by the prions? This causes neurodegenerative disease, and in cattle you could see this is a mad cow disease, and humans we call it as a Creutzfeldt-Jakob syndrome. These are the examples that you could see in this case actually. So we're done with almost the subviral agents and scrapie. Yes, exactly. We call it as a scrapie disease that usually it get affected by means of prions. We call it as scrapie, and we call it screw disease. CJD. See this CJ Creutzfeldt-Jakob disease. CJD. This is otherwise called as Kuru disease. CJ Creutzfeldt-Jakob disease we call it as BSC. BSC is like bovine spongiform encephalopathy. BSC, which stands for bovine, yeah, spongiform encephalopathy. This we call it as mad cow disease. This mad cow disease can be otherwise called as bovine spongiform encephalopathy. So from this area, you can get a questions, especially with the prions. So we almost be done with this part. I mean, the virus. Only thing remaining is we need to discuss about corona. We have to discuss about coronavirus, cultivation of viruses, diagnostic perspective, and almost we discussed about the pathogenicity and negative points of viruses. We discussed so many things about negative. Let us see in next class about the positive aspects of the virus. There are some beneficial role played by the virus also. In the next session, we'll discuss about the coronavirus and the cultivation of viruses, and also we discussed about the diagnosis, how we can diagnose if the person seems to be confirmed with viral diseases. What are the diagnostic techniques that we'll do, like PCR, Western blotting, ELISA, serological test. There are different types of tests, advanced technique, future perspective studies. Like these are the things that we're going to discuss, because this 2 hours, like in which I thought of completing this particular part, that is virus, maybe I couldn't complete. Maybe in the next class we'll discuss about corona. It's a life cycle of corona that we'll discuss in the next class, and we discuss about the other parts, the cultivation. So this is the diagnosis. What are the different diagnostic methods for most of the viruses that we'll discuss in the next class? There we'll discuss about PCR also. PCR, what are the NAS by SBA, RPA, all these type of different techniques that will be microarray technique that we'll discuss. And how do we diagnose corona patients? This we'll discuss in the next class of corona, because the season for corona, we'll discuss about how we diagnose the corona by means of RT-PCR that we'll discuss. Tissue culture technique, how we can cultivate the virus, cell culture, expand culture, and this is about cell lines. By means of cell lines, how do we do it? And egg inoculation. By egg inoculation also we'll discuss, because from this area you can get a question. So these are the methods of cultivation of virus. Only these parts we seems to be pending. We are in biological classification. Tomorrow we'll discuss or complete the virus, and then we'll start with the bacteria. So I hope you're done with the session, and yeah, tomorrow we'll completely go with the corona and complete, like case studies and all the things we discussed about corona and the cultivation of virus. So thank you for joining with us. So hope you paid an attention and practice all the test papers. We'll meet you tomorrow. Thank you so much.